Okay, let's continue. The next presentation will be about mercury analysis in coal and related task in stack gas of the coal-fired power plants. And what is the problem? So, as it was shown by assessment made by UNIDO, mercury emission from the stationary sources like coal-fired power plants are one of the most important sources of the mercury emission to the atmosphere. And so according to estimations, coal combustion gives about 25% of global mercury emissions. And main problem is here that in all the world, coal-fired power plants are very different. So there are some modern stations which are quite effective in mercury removal from the stack, and there are old ones. And trouble here is to make assessment of all available stations and to understand what particular measures should be done for particular stations to reduce mercury emission from coal combustion. And why is this problem so important? Because it was found that in coal always some mercury is present. So here you can see some information that this concentration in mercury in coal can vary from very low, from about a few ppbs, to very high, about hundreds ppms. So more than 5 orders of magnitude is the variation of mercury concentration in the coal. And average mercury concentration estimated based on about 100,000 analysis is about 100 ppb. And all the modeling, all the estimations of mercury emissions from coal-fired power plants are made based on this value, which is really not so precise, and I will show you why. And what is important to understand, that there is no obvious difference in mercury concentration depending on the type of the coal. So different types of the coal can show you different patterns. Yeah, and you can, there is no evident connection between type of the coal and mercury content. And here initial syngenetic mercury content, so it's due to its formation, is really quite low. It's from 5 to 40 ppbs. And it's just comparable to what we can find in the plants and peat. The problem is that the deposits, coal basins, are affected by hydrothermal processes, and this is causing mercury enrichment in the coal deposits due to this process, and also causing variability, which we can see. And why all this happening? So here you can see so-called global mercury belts. Yeah, so this one, and here this one. The last one is most important for you. And with red, you can see the most important coal basins around the world. And with black dots, just known mercury deposition, most important mercury depositions. And what we can see, that in many cases, coal deposits are overlapping with so-called global mercury belts, which means that mercury deposits are on beneath of the coal basins. And mercury is just going up in the earth core and going finally to the coal deposits, and so enriching it. So mercury is soaked inside the coal basin. And this is why we have in different parts of the world very different concentrations. So here, for example, Russia, which has one of the most richest part of the world deposits of coal, you can see that most part is outside of the global mercury belt. And in Russia, mercury concentration in coal is quite low, except this part, which is really also in global mercury belt, in Eurasian belt. And here mercury concentration found also is quite high. And also there is a problem. And here you can see the analytical data which we are using for just all the modeling and all the calculations, yeah, all the modeling of the emissions and so on around the world. And the most important work here is estimation of the mercury concentration in the coals all around the world. About 40,000 samples they analyzed, and about 100 ppb was found, but with very high variation. You can see here it's work of Curtis and all. In the United States it was done quite comprehensive study of variation of the mercury in coals, and you can see about 40,000 samples were analyzed, and reported, really data reported are a little bit different, but according to a report to Congress made in 2002, average mercury concentration in coal in the United States also is about 100. But also you can see very significant variation depending on the source. In other parts of the world, unfortunately, assessment made it's quite doesn't have quite enough quality of data. And for example, in China, you can see not so many samples were analyzed in different time, different researchers. It was official report made by UNEP, but without really many details. And in Russia, quite many samples were analyzed. In other parts of the world, here you can see that only a few data available, and so it's very difficult to estimate what's the real situation with the mercury in the coal in these countries. And so for them it's unclear, so all the estimations are quite uncertain. All the emissions for Australia, Indonesia, for example, quite uncertain. And this means that we need to receive more data about mercury content in different types of the coal around the world, and this is what many researchers are focusing on. And how to do this? We are providing this tool, universal tool for coal analysis and related, so fly ash analysis and so on, which is important for understanding what's going on during coal combustion and also what's going on in the stack. It's the same procedure. So we are following standard method. This is ASTM D6722 method, which is describing using of a thermal decomposition method for coal analysis. So we are talking about 700 degrees Celsius in the furnace during analysis, and so at these temperatures all the species, mercury species present in the coal are decomposing, giving you atomic mercury, which we can detect. But for industry, it's very important to understand what real species we have in the coal. So because depending on this information, you can treat coal before combustion in a different manner, and also you can plan the amount of reagent which you will need to bind the atomic mercury to ionic in the stack. And to do this, we have introduced special thermoscanning method. And what is the general idea? That mercury compounds are all unstable towards heating, but they have different temperatures of decomposition. And so you can separate different mercury species according to different temperatures of their decomposition. And so the principle of the method is registration of the dynamics of the mercury release during gradual heating of a sample. Of course, there are some shortcomings, and we have to understand that thermochemistry can be quite rich, yeah, especially in the presence of oxygen and so on. Nevertheless, for charcoal, for coal combustion, for example, pictures which we can retrieve using these methods are quite interesting and gives a lot of information. For example, so here thermospectra of 2 bituminous coals from different regions in South Africa, and you know this is one of the big exporter of the coal, and one coal is in blue, it's with quite high concentration, about 500 ppb, and another one in red, lower concentration, about 200 ppb. And you can see that the thermogram of these two coal is very different. So for first coal, blue one, you can see that almost all the mercury is released at quite low temperatures. At about 200 degrees Celsius already is enough to release most part of mercury from this coal. And this peak can be attributed to atomic mercury soaked on the crystalline structure of the coal. It's not chemically bounded. And additional peaks can be attributed to the presence of this one to mercury chloride and this one to mercury sulfide. But you can see much less ratio of these two mercury species. For second coal, you can see that not much metallic mercury was found in the sample, but main peaks are attributed to mercury chloride and mercury sulfide. And presence, for example, of mercury sulfide can be proved by appearance of SO2 in the gas flow at temperatures starting from about 500 degrees Celsius, with maximum at about 600. So it's at the same time you can see the presence of SO2 in the flow. So here you can see quite clear pictures, so you can attribute different peaks to different species, and really this is very important. So very important for understanding of treatment of the coal before combustion, and also how to deal with stack gas. So what species you will have there, what will be the ratio of these species, so how much of the brominated coal, for example, you have to inject inside the stack to bound all the atomic mercury to ionic form, and how efficient will be the MRU unit on this particular station. So depending on what principle is used in these units. Also, what other applications you can imagine? So for example, for demercurization, it's very important to understand what species do you have. And if you are doing remediation, so you can use different temperature for thermodistillation of the mercury from soil, for example. So you can use all the time 800 degrees Celsius, and you can use a lot of electricity to do this, and you will have to pay a lot for just to solve this task. But if you will run this thermoscanning, and if you will find out that most part of the mercury present in the contaminated soil is just not bounded atomic mercury, so you can use much lower temperatures for thermodistillation. So for example, 400 degrees Celsius, and it will be enough, and you will save a lot of money just doing the same, with the same result. And related task, so when we already have prepared the coal for combustion, so we have combusted it, and we have the result, we have the stack gas, and we have to control mercury in stack gas to control the efficiency of mercury removal from the stack, and so on. And how to do this? And US EPA has introduced special method 30, and originally it was only online monitoring, so which is now called method 30A. Idea was that all should be fully automated, and of course, if it's possible, so it's just a great advantage. So real-time response and so on. But it happened that the cost of installation of such systems and running cost of such systems appeared to be really high. And it was very difficult for industry, and also one more problem has arisen, that it was difficult to validate the continuous emission monitors because it was no tool how to validate it. So what you see as the result of monitoring, is it related to reality somehow, or it's absolutely senseless. And so to solve all this trouble, US EPA has developed special method 30B, so-called sorbent trap method. And US EPA considered it like reference method, and main idea was to decrease installation cost and running cost. And here you can see what's the difference. Difference is that SEM gives you all the variation of the mercury during the time time of observation. Sorbent trap method gives you average. Yeah, so just how much was emitted for this particular time of sampling. But it's what is specific for the industry. You don't need to report data for every minute, every hour. You have to report weekly average, 2 weeks average, and so on. But you must do it with good efficiency and preferably with low cost. And this is exactly the reason why US EPA Mercury Toolkit was developed. So just the special tool which can be used for validation of the continuous emission monitors, and as well to provide testing of different stacks, including long-term monitoring of the stacks as well. And so this special kit was initially developed in the United States, but in recent years it was tested in different countries around the world, and it was introduced for different applications. So just in Russia, China, South Africa, Southeast Asia, and so on. And here you can see official booklet of US EPA, and our instrument is listed here, yeah, as the part of the official Mercury Toolkit. And I will explain why. So this is what this US EPA Toolkit consists of. So it should be sampling system, so different sorbent traps. There are standard traps and there are some special traps for different applications, and analytical system for analysis of the traps. And sampling system can be different. This one is which was developed by our joint company Ohio Lumex in the United States. But there are enough manufacturers on the market, present on the market, and you can purchase any. Sorbent traps also were originally developed by Ohio Lumex in cooperation with US EPA, but now are available from different manufacturers as well. But analytical system, recommended analytical system, is available only from us. And the reason is quite obvious, because golden trap-based analysis cannot deal with high mercury loads. And here, normally, especially when we are talking about not about classical method 30B application, but about long-term monitoring, so the upload of the mercury can be tenths of micrograms on one trap. And so this is too much for golden trap-based analysis. So that golden trap will be just destroyed after analysis. And here you can see different designs of the sorbent traps. And what is the idea? Here we have classical method 30B sorbent trap. It has two sections. It's activated charcoal-based sorbent with some treatment, like little brominated. And first section is analytical one, and gas flow is coming from just this direction. And all the mercury species should be sorbed here. But you have breakthrough section to check that mercury is not escaping sorption, not escaping analytical section. And as a result of the analysis, you have to find out that all mercury is trapped here and less than 5% mercury is here. So then result is correct. Also, special traps were developed which are used for long-term monitoring, but also for initial testing of the stack, just to understand how composition of the stack is affecting the sorption efficiency of the mercury. And they have additional section, so-called PS12B method trap. And this additional section just has the mercury spike. So here there is the known mass of mercury, and the idea of the analysis that after sampling you have to analyze all three sections, and you have to find on the spiked section the assigned value of the mercury. Why it's important? Because when we are talking about method 30B, sampling time is quite low. It's about half an hour, usually. When we are talking about modification of the method, so which was Appendix K, now it's separate method, PS12B, so it's already long-term story, about a week, 2 weeks, and you can imagine the stack. In the stack we have a lot of acidic gases, SOx, NOx, and if we are talking about short period of time, we are not affected seriously the surface of charcoal. But if it's about long-term monitoring, so nobody knows how this particular stack gas composition will affect sorption efficiency. And you can find what? That with the standard trap, there is no mercury in the analytical section, there is no mercury in the breakthrough section. But it's not definitely means that it was no mercury in the stack. So it can be just breakthrough of the mercury from both sections, just because high concentration of acidic gases in the stack. So initially mercury was sorbed, but then it was kind of eluted with acidic gases. And you have just lost it. And to be aware of such situation, we have, like the last section, we spiked mercury trap. And if we will see any loss of mercury mass in this last section, it will exactly means that we have this difficult situation, that we have to do something, so we have to customize design of our trap to reduce the effect of acidic gases. And there are such kind of traps with special screw-bars for acidic gases just to reduce this effect. And during the initial installation, this is what should be done. So conditions should be optimized in design of a sorbent trap also should be determined. And also it's possible to use speciation during sampling. So we can use special speciation traps, and they have two different parts. One part is responsible for sorption of ionic mercury. So here we have potassium chloride, also analytical section and breakthrough section. And ionic mercury will be trapped here. All other species, atomic mercury, mostly of course, will go to the next section to carbon trap and will be sorbed here. So you will have separation during sampling, and this is also very important for just understanding of the efficiency of the mercury removal from the stack. So also a lot of analysis are done using these speciation traps. So here just some comparison of different approaches. So here the picture of the probe itself. What is the probe? The probe is the long train, about 2 meters long, just to have an option to install it to any stack. So we have to reach about the central part of the stack, so it should be long. And also you can see here that at least 2 traps should be sampled in the same time. So here there is built-in quality control. So you are analyzing at least 2 traps, in some sampling system 4 traps, and they can be standard FATB trap and another standard FATB trap, or it can be combination FATB, PS12B traps, just to control everything. And anyway, as a result, you have to present results of analysis of 2 traps, and difference between them should be less than 10%. If it's more, so it was the problem with sampling, so it was not correct. And here are some results of breakthrough studies. So different client traps, about 600 of them they analyzed, and here you can see that all the breakthrough is less than 5% mostly. Yeah, so all the mercury was trapped on the analytical part. And here the same study for spike recovery. Tolerance limits plus minus 25%. But you can see that most all the results are in 10% range. Yeah, so recovery of the spikes is quite good. And this testing was done in United States in a lot of different coal-fired power plants. And you can see very nice results. And this is why this method was adopted, and now it's starting to spread around the world like parts of national legislation in other countries. And this method also was standardized by UNEP and is available on our official site. This method can be used for the countries which still don't have their national method, official method. So they can use UNEP procedure. And then we were dealing with sorbent traps, so we faced with the problem that sometimes nobody knows how much is the mercury load on the trap. So we had the solution for high mercury concentrations, like it was special mode, and temperature profile was just gradual heating of the sample, just not allowed to mercury to be released in one shot, just to postpone mercury release to spread it in time. Yeah, soon. But for sorbent traps, especially for method PS12B, it was not enough. And so we developed just closed loop control system, which is just interfering to the programmed temperature profile, just to keep signal in the range, just not to allow it to escape to the range of saturation, non-linearity, and so on. And we can control the attachment using signal or background absorption is the criterion. And how it works. So here you can see different profiles of mercury release, different dynamics, depending on the mercury load on the sample. And for lower loads here, about 40 nanograms, so no problem. So just it's released, that's all. The higher is the load that we can see, the more complicated the shape is becoming. And with highest shown load, 40 micrograms here, it's shown why, so we can understand it. So here we have special parameters controlling how signal is changing during analysis. So we have 2 preset levels of the signal, and if during analysis signal is going higher, to values higher than the first threshold. Software is intervening the temperature profile, and it's stopping heating temporarily, so just pausing the temperature profile, yeah, and it's operating itself. And if signal is going down, so it's allowing to continue, and it's so controlling. And here, for example, you can see a result of such control. So this kind of, sinusoid is the control. So the system is just maintaining the signal on this level, just not allowing to increase too fast. Yeah, we have one more parameter is just controlling how fast signal is increasing. If it's increasing too fast, system is not waiting when it will be already close to second threshold, just stopping heating, not allowing to continue heating. And here you can see it's heating up, cooling down, heating up, cooling down, yeah, until it will be fine from the point of view of the signal value and the rate of the increase of the signal. So it's allowing to just finalize here the analysis, yeah, and that's all. So it costs time. So when we are dealing with high mercury loads, it takes more time because system is just postponing mercury release. It's just distributing it in time, taking more time to release all the mercury. Finally, it finalizing the original temperature profile, so it's just providing the highest programmed temperature, but the rate of temperature increase can be very different depending on the load. And here you can see what's the difference between low and high loads of mercury. So if it's not so much, so just we are using standard temperature profile, but if something happens, system is intervening and controlling itself. And this is very important for exactly sorbent traps, because at any cost we have always to deliver the result of the analysis. It's not possible just to say that's not good result. It must be delivered. If not, it's fine. Yeah, so and it's quite costly for the plant. And here, just analysis of the coal and how it works. So if we have the coal with known mercury content, in this case it's 20 microgram, it's amount of the mercury in the sample, yeah, which we are analyzing. And if we are doing analysis without this control system, what's going on? We can see that during analysis we have this green line, it's our quality line, is rising rapidly, and it's just going upper than our threshold. So for us, green line less than 2, it's fine. If it's more, it's already a situation when we can have trouble with correction of a signal. Why? Because level of a green line 2, it means that optical density of a sample inside the cell, it's more than 2, and which is equal to less than 5% of the light is passing through the system. So it's almost all light is absorbed. So it's it's not precise correction in this case, and even some artifacts could happen if light just not penetrating the cell. And in this case, what we see this profile of the mercury release, but it's false profile. So it's this negative peak really should be positive. So we are losing signal here because light could not penetrate the cell, and Zeeman is not normally correcting the signal. So software will give you the message that your result is not correct, but it will not help you already, yeah, if you have like only one sample of this type, yeah, like sorbent trap. If you can just take another sample, no problem. But if it's sorbent trap, no, it's unique. And here you can see instead of 20 micrograms, the measured value is about 10. It's about 10 here. Yeah. And the same analysis, but using this control system. And you can see here just cycle of heating up, cooling down, heating up, cooling down. And green line, it's all the time is kept on the normal level, just not allowing to give too much optical density inside the analytical cell. And so the result of the analysis in this case, by cost of some additional time, not so much really here, it's much closer to the value which must be. It's 19.9 microgram, the load of the mercury in on this trap. And here are some examples. So a lot of installations in the United States, in China, and a growing number of installations in other countries. And moreover, this is installations just on coal-fired power plants. But these methods, it's spreading to other applications. So it's now used to control the mercury emissions from stacks from different smelters. For example, here, one of the smelters in Spain. Also, this method is used. Also, more and more attention to cement production sites, and also the same sorbent trap technology is used to control them. And really, this is all for this presentation. Yeah, so do you have any questions?